Welcome to the second installment of our Thrips Identification video series. In this video, we'll go over the different thrips life stages, the anatomical features that we use when IDing thrips, and how to tell the difference between male and female thrips. Let's start by familiarizing ourselves with the thrips life cycle. Most of the species we encounter in greenhouse crops lay their eggs inside the leaf tissue. After a few days, the egg hatches into their first developmental stage, which we call a first instar larva. This stage only lasts 1 or 2 days, before turning into the larger second instar larva. After 3 or 4 days of feeding, many species make their way into the substrate or other concealed location to pupate. After the pupal stage, they will emerge as adults to start the cycle all over again. This process takes approximately 2 weeks for most common thrips species, although this varies a lot based on temperature, host plant, and thrips species. When you're sampling thrips directly from the crop, especially if you're doing plant taps, you're likely to get a mix of immature and adult thrips. The first and second instar larvae have long, squishy bodies, and they're usually some shade of yellow, beige, or white. They have tiny eyes and no wings. You may also see green stomach contents through their translucent bodies. There are 2 stages of pupae. The prepupae look just like larvae, but they have tiny wing buds. The pupae is when they start to look more like adult thrips. Their eyes are larger, but they now have wing buds that are much longer. Like the larvae, they are still usually pale and squishy. Their antennae are also folded back over their heads. Once they reach adulthood, they are usually more pigmented, most have long fringed wings, and their antennae are not slicked back. When we're trying to determine thrip species, note that you can only use adult thrips and you can't use any of the immature stages. Our grower key only uses features that can be seen in adult thrips. Insect bodies are divided up into 3 segments: the head, the thorax, and the abdomen. In thrips, the thorax is further divided into segments: the pronotum, and the mesonotum and metanotum. The pronotum is distinctly separate from the meso and metanotum. Thrips' wings are unusual compared to other insects. They are made up of narrow, translucent blades that are fringed with long hairs. Although taxonomists often spread their wings out wide like this when they're identifying them, live thrips typically hold their wings flat against their back. If you have collected thrip specimens in alcohol, their wings might drift out to the side or remain held flat onto the back, which can sometimes make them challenging to see. Not all features are visible from all angles. When you get the thrips under the microscope, what position you need to arrange them in depends on what you're trying to see. When we're identifying species, we need to look at them with their wings facing towards us. When we're trying to determine if they're male or female, we need them facing the other way so that their legs and beak are facing towards us. Let's first look at female thrips. The main identifying feature is the ovipositor. This is what the female uses to insert her eggs into the leaf tissue. If the ovipositor is flat against her abdomen, it will look like a double fish hook when the specimen is face up. If the specimen has been stored in alcohol, the ovipositor might be extended, and you can see this by flipping the specimen onto its side. You might also notice a single orange dot in the abdomen just above the ovipositor. The next feature we can look at is the abdomen's shape. In species where the abdomen is very dark, this might be the only feature that we can see. The female abdomen is usually wider than the thorax, and then it tapers to a point at the end of the abdomen. Male thrips are generally smaller and paler than their female counterparts. Instead of an ovipositor, the male's reproductive organs is this orangey-yellow jelly-like structure. There are 2 rounder blobs connected by a thinner strand. The abdomen is skinny and is not noticeably wider in the middle. The end is blunt and rounded. To put it simply, the male's abdomen is shaped like a hot dog. Why would a grower want to know the difference between males and females? The main reason for this is sexual dimorphism, which is when males and females look very different from each other. Think of male and female cardinals, for example. The same is true with some Thrips species. For example, in Thrips parvispinus, the males are pale yellow and the females are dark brown. At first glance, you could mistake them for two different species. There are other dark-colored Thrips species where the males are also dark, so being able to spot sexual dimorphism can be useful for identification. There are some Thrips, however, that do not look like either the males or females that I just showed you. These are the aptly named tubulifera. The tubulifera is a suborder of Thrips whose most notable feature is that the last segment of their abdomen is an elongated tube. The females of these species do not have a stabby ovipositor, and instead they lay their eggs on the leaf surface. These are pretty uncommon as crop pests, and most growers are unlikely to ever encounter them. The hollyhock thrips and the weeping fig thrips are a couple of species of tubulifera that some ornamental growers might encounter, although they're not very common. Now that you're familiar with basic Thrips anatomy, in our next video we'll go over how to identify Thrips using a dichotomous key. Visit the On Floriculture blog for a copy of the Thrips key and more identification resources.